Yes. Okay. So, good morning to all of you. After a gap of 1 week with your exams, we are back to the basics. And for your memory, I would remind you that we are talking, discussing, debating on the chapter Molecular Basis of Inheritance. And I am happy that some of you have practiced the structures of each chemical molecule, and some are still in the process. So those who haven't done it, please do it, because you got the experience now. I have sent the model paper for term 1 proposed by CBSE in the month of November, December. You must have gone through that question paper. If you are not yet, please go through it. I have already posted in our WhatsApp group, and you would be seeing many diagrams. It's called diagram based questions. Many diagrams are there. In fact, very few questions are there without diagram. Please go through that, and invariably you will find that a diagram will be there, and somewhere there is a mistake in the diagram, or somewhere the labeling is missing. You are asked to identify it. So unless and until you practice the diagrams, term 1 exam will be very difficult for you. So let us make it simple and easy, and the best way to do it is practice, practice and practice. Whenever you are bored of reading something, studying something, take a pencil, start drawing the diagram. And that's a good skill. Drawing a diagram in a beautiful way, scientific way, is a good skill. So you can develop that skill. Like already you must have practiced the double helix model of DNA. You must have practiced the pentose sugar molecule, right? Phosphate molecule, and all the nitrogenous bases, purines as well as pyrimidines. And once you start practicing it, now you must construct a DNA ladder by joining them, all the 3 molecules, right? We said SP. What is that SP? Sugar phosphate, isn't it? Sugar phosphate, SP. So this S and P makes what I said, vertical structure of DNA. All these molecules, of course, you have practiced, and here comes the sugar molecule. I hope all of you have practiced at least once, and you will be familiar with the carbon positions. When we say pentose, penta, 5 carbon atoms, there is a tendency in you that you may label carbon in the O position, oxygen position. Because it is a pentose sugar, there is a chance. So to avoid that error, look at that. You need to practice this first, pentose sugar. Wherever that numbering is there, no? 1, 2, 3, 4, 5, they are carbon atoms. So you can write C, 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 C. So first carbon, second carbon, third carbon, fourth carbon, and fifth carbon. And centrally there is O, oxygen. So pentose sugar. And pentose sugar is attaching itself to nitrogen base, where at first carbon. This is important. To which carbon? Another molecule is attaching. And what is the bond that is formed? N-glycosidic bond. And then once you write nitrogen base, now you need to realize this structure. Is it a double ringed structure or single ringed structure? If it is purine, already I told you it is a double ringed structure. You need to practice like this. There in pentose sugar, you are labeling it as carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, penta 5. Here it is nitrogen. That's why we are calling it as, Vivek, nitrogenous bases. Isn't it? Now look at the purines, adenine and guanine. And how to remember this in the exam? I said gap. GAP, gap. G for guanine, A for adenine, P for purines. Correct, no? This is how you have to remember. And if gap is there, what is next? Pyrimidines. So cytosine, thymine, and one extra has come here, uracil. And remember, I told you uracil is present in RNA, where in place of thymine. That's why we are writing side by side, thymine and uracil. Now here when you are taking a pencil and drawing these diagrams, you must put position of nitrogen. How many nitrogen atoms are there? Where are you putting these hydrogen atoms? And what is the functional group in that? See, your chemistry teacher must have told you functional group. So you have to place functional groups. Now, this nitrogenous bases, adenine, guanine, cytosine, thymine, uracil. These are to be linked with the sugar molecule. Okay? And remember we said nucleoside and nucleotide. If someone asks our Sandeep, what is DNA? Sandeep says, DNA is the secret of our life. But if someone asks Vivek Kadkol, what is DNA? You will say, polynucleotidal chain. Okay? If someone asks Rakesh, Rakesh will tell, Jai Jagannath. Our secret is our Jai Jagannath. So, here, when we say the DNA is polynucleotidal chain, what do you mean by that? Polynucleotide. Poly means many. Now you must understand what is nucleotide. To understand that nucleotide, you must know what is nucleoside. Okay? So what is nucleoside? As you are seeing, a pentose sugar is linked to nitrogenous base. That is nucleoside. And in my previous class I told you how to remember this. What is nucleoside? Many times in your UPSC ND exam this will be asked. The difference between nucleoside and nucleotide. So see the spelling, nucleoside. In the spelling, N and S is there. N for nitrogenous base, S for sugar. So NS nucleoside is equal to SN, sugar plus nitrogen base. So nucleoside plus what is left out in the DNA molecule? Phosphate group. So nucleoside plus phosphate is nucleotide. So all the three molecules have come. Sugar molecule, nitrogenous base and phosphate molecule, NSP, that is nucleotide. If many are there, such structures, nucleosides and nucleotides, and that is the reason why Kadkol says it is a poly, means many, nucleotidal means nucleotides, chain, linking with each other. Look at that, nucleosides which will be present in DNA and RNA. Already we have discussed about this. Okay? Just for your memory sake, I am repeating this. Right? This is how the nucleosides will be there. And there comes the nucleotide. Look at that. Now I want you to understand the linkage. I said to first carbon, nitrogen base is attached. And the fifth carbon, phosphate group is attached. Okay? First and fifth. Since all the three molecules are present, sugar molecule present, nitrogen base is there, phosphate is there, it's a nucleotide. So now it is easy for Kadkol. If someone asks what is DNA, polynucleotidal chain. And we said just now glycosidic bond. Now we have got another bond, phosphodiester bond. So this phosphodiester bond, as you are seeing, is present where? In the phosphate group. Okay? Now you can see here The nucleotide has an additional hydroxyl group, OH group, as you are seeing here. Look at that. So, in the second carbon position, there is OH group. It is rounded off, you see, with red color, OH. Whereas in DNA, the same sugar molecule will be losing that oxygen atom. That is the reason why we say deoxyribose. D means minus. Minus of what? Minus of oxygen atom. So deoxyribose. Whereas in RNA, it is oxygen is very much present in the form of hydroxyl group at second carbon position. Hence we say ribulose or ribose. So the bond that is attaching the sugar with another sugar through the phosphate molecule, hence we call it as diphosphate bond. Now, for all this research, whatever we are talking it and taking it easy now, is being made easy because of the hard work and contribution of the following great personalities. And their hard work and their success. Remember, always hard work pays the success, and success leads to recognition. And these have been recognized by the world's greatest awards, Nobel Prize. Okay? Frederick Mesher identified DNA and named it as nuclein. And of course, our favorite, Professor Watson and Professor Crick, proposed the double helix model, for which they got Nobel Prize. And then we are thankful to Wilkins and Professor Franklin for X-ray diffraction. So we understood now what the DNA is all about. So basically, since we say DNA is like a ladder, I said Nichenige, all your houses have got that ladder. You climb on the ladder and reach the top of your house, Hosla house, Udayar house, Chaliki house, like that. So you hold on one side, another your friend hold on the other side, and then twist it. What you get, that is the helix. Since there are two stripes, we say double helix. That's why we say DNA has got double helix. And this double helix model is proposed by Watson and Crick. So DNA is made up of two polynucleotide chains coiled in right-handed fashion. The twist is towards the right direction. And the pitch of the helix is 3.4 nanometers. Number of base pairs in each turn is 10. What is that base pair? Nitrogenous base pairs. And already I told you that base pair ruling. What is that? One purine combines with another pyrimidine. Now this is how the thing will be. Look, this is the base pairing rule. Adenine always combines with thymine. Guanine always combines with cytosine and vice versa. Thymine T can also combine with A, adenine. Cytosine C can also combine with guanine. So the base pair rule is fixed, and you must remember that the nitrogenous bases will pair in a particular fashion. And when we say 3' to 5', it's the carbon atom at which it is attached. And that is why we say in the properties of DNA, we say anti-parallel. Just like railway lines, the ladder is vertical strands are just like railway lines, but then polarity is different. One thread starts from third carbon, so we say 3', ends with 5', for fifth carbon. Another starts with fifth carbon and ends with third carbon. So we say anti-parallel. So this is how the two professors, Watson and Crick, identified DNA through various experiments. Okay, and now further the bonding is shown to you, how sugar molecule is attached to nitrogenous base. Is taken cytosine, for example, sugar and cytosine, and that fifth carbon is attached to phosphorus. So you can see there is presence of three bonds: a glycosidic bond, a diphosphate bond, and between two nitrogenous base pairs there will be hydrogen, right? Somebody is waiting because, okay, so there will be weak hydrogen bond, and thanks to that, sometimes it is good to be weak. If it is weak, since it is weak, we can just supply a little bit of energy, the hydrogen bond will be broken. So the two threads which are attached up to now will be divided, and we call this as replication, DNA replication. And once DNA gets replicated, it is easy for us to create varieties. So thanks to weak hydrogen bond, had it been strong, maybe things would have been difficult for us, right? So further, you can see the attachment phosphate group. Yeah, bond between two nitrogenous bases is weak hydrogen bond. Because hydrogen bond is weak, just little bit of energy you supply, hydrogen bond will be broken, as I am showing in the picture. You can see the breaking of the hydrogen bond between two nitrogenous bases. That results in DNA replication. Same thing we can see here. Look at that. I hope you are able to understand here base pairs, how they are being broken. And once that breaking takes place, DNA replication occurs. That results in creation of varieties. And of course, there is just like in chemistry, you got Chargaff's rule. It is the proportion adenine plus guanine is equal to thymine plus cytosine, right? And that should be equivalent to 1. That is the in terms of what we call density, okay? And then further, exactly in the form of molecular way, how to represent, this is the way we represent. Now you can understand, see the left side structure and right side structure. Then I am sure you will understand 3' and 5' in one side, in another vertical thread 5' and 3'. That makes DNA anti-parallel. Okay, all this we have covered already, but still just for your memory sake I am repeating it. And then the same thing in terms of calculations is proved by X-ray diffraction, and number of base pairs easily calculated. And now all these things, now the question comes, if the DNA is so tall, lengthy, and remember, where is DNA? It's inside the chromosomes. Where are the chromosomes? Inside the nucleus. And how big is the nucleus? All of us know, it's a small one. Particularly in prokaryotes, in bacteria, there is no nucleus at all of you know, there is only nucleoid, because there is no outer cell membrane. Already you know in junior classes. When it is nucleus without nuclear membrane, it is nucleoid. And you cannot differentiate. Sir, yeah. Yes,
sir,
please.
then DNA in prokaryotic cells will be present in nucleoid, sir?
Yes. In fact, you can say cytoplasm. You can say DNA is present in cytoplasm, because we cannot distinguish which is nucleoplasm, which is cytoplasm. Everything is protoplasm only. And already you know in junior classes that in all the prokaryotes there won't be any membrane even in the cell organelles. Mitochondria, plastids, Golgi body, ribosomes, these are membraneless. There is no well-built membrane. Everything is like our science school swimming pool. You cannot distinguish, right? So, how is that this DNA molecule will be present there? Okay, in prokaryotes, like in bacteria, E. coli, the DNA is not scattered throughout the cell. It is restricted to one area. That is the reason why we are calling it as nucleoid. It's not spreading across. That's the meaning. And it is negatively charged, so it is held with some positively charged proteins to form nucleoid. So if someone asks you what is nucleoid, it is DNA negatively charged attracted by positively charged proteins. So you can say nucleotide is equal to proteins plus DNA, right? How about in eukaryotes, in us? How this DNA will be packaged? As you are seeing here, I'll just let me expand this. Look at that. Yeah, this is how it will be. We say nucleosome. So there is a protein. Surrounding that protein, DNA molecule is there. Okay, and in specific, it is called histone. That protein. How many? 8 histones will be there. So we say octamer. So this is how the DNA package material will be present in all the eukaryotes. Whereas in prokaryotes,
This histone DNA.
yeah, histone is a protein. Histone is a protein. Okay, centrally there is a protein molecule, that too 8 in numbers, 8 histones in like in the form of a hockey ball. And when you take the hockey ball and put the thread around it, that thread will be DNA, that hockey ball is protein. So this is how the DNA molecule in eukaryotes will be present, right? So you can see in eukaryotes there is a set of positively charged, okay, base proteins. Now you can see here those 8 histones. Let me expand. It's called octamer. Look at that. Okay. H for histone.
Yes.
So inside, I said you take hockey ball. That's a protein, spherical shape. To that you tie some thread. That thread is DNA, and that hockey ball is protein. And within the hockey ball, again inside, there will be pebbles, round pebbles. H1, H2, H3, H4, like that, H8. That is octamer, histone octamer. Hope clear? Now, when we stain it, how it looks like? This is how it looks like. Okay? 8 molecules of octamers, octohistamides, covered by the DNA. And that's why we say nucleosome molecule. Right, let me allow Anurag. Anurag is waiting. Right. Right. Okay, so we have got 3 varieties of this. Look at that. And when we further magnify it, you can see a typical nucleosome contains 200 pp means base pairs. So total number of nucleosomes in us will be 6.6 into 10 to the power 9 base pairs by 200. Approximately is giving that DNA length 3.3 into 10 to the power 7.
3.3 into 10 to the power
7. How is that possible? This is one molecule we are talking of, right? So look at that. Further, from top view, and actually the linkage will be like this: one nucleosome to another nucleosome. The linker DNA, that's an enzyme. Again,
Sir, what is that minus 10 nanometer, sir? Is it
which one?
minus 10 nm, sir, at right
Right.
of nucleosome?
Yeah, that is nanometer is the distance between the protein molecule and the DNA, the DNA thread. Okay, that is that. That means you must appreciate the minute things also are being studied in case of human DNA. All this will come under Human Genome Project, HGP. Okay, we will be talking about this at length in chapter number 12, biotechnology, Human Genome Project, right?
Yes.
Further you can see nucleosome constitute the repeating units to form chromatin. Now what we are talking is, what is DNA? inside DNA, what are the chemicals that are present, how a nucleosome will be formed, and nucleosome to chromatin material to chromosome. I hope you are able to understand. Because we said, where is DNA? DNA is present inside the chromosome, we said. That means, ultimately, the DNA should be packed in such a way that finally it should appear like a chromosome. And already we said, chromosome will have two threads, chromatids, and a central bead, rudraksha I said. You can see here. That is the structure of a chromosome. Now, you will appreciate how this chromatin material is. Look at that. This is a chromosome. Inside chromosome, those coils are there. They are called supercoils. And when you highlight this coil, inside small coils will be there. And further when you highlight this, you will see the nucleosome. And further when you break this nucleosome, what you will see? That histones. And all these in a joint together, you are getting DNA. Okay, Rakesh? I hope you appreciate
Yes, sir.
the research work that is done. Okay? And this is what I asked Sandeep, Vivek, Rakesh, Prasad. You must also do like this. That will give you a lot of maja for you. Plus, it's a great contribution to humanity. Many diseases are coming. We were not knowing, see, why people suffer with particular diseases. Unless and until we understand the structure of DNA, we understand the structure and position of the gene, we were not able to know why people suffer. So that is the reason why we should be thankful to Professor Watson and Crick. See the work that they have done. And remember, Rakesh, this work is done in 1963. Now Rakesh is almost coming to 2023. So we need to upgrade ourselves. What happened after that? That is the reason why I always tell our Kadkol, go through the scientific magazines, Young Scientist, Nature, okay? And you must become friendly to great scientists through LinkedIn. You must become friendly so that you upgrade yourself, what's happening. And further, you can see the beads, that rudraksha, right? Let me expand this. How chromosomes are appearing. Now, see, chromatin is packaged. What is that chromatin package? Just now I said, octanes. There are 8 histones and they are rounded off with a thread. That is chromatin package. Protein plus, what you said? DNA. And chromatin fibers are formed. Those are in the form of a spring, coiled and condensed at metaphase stage, and that results in chromosomes. So this is how inside the chromosome DNA material will be present. And higher level packaging of chromatin requires non-histochromosomal proteins. Okay, further you want, then we don't require this histone, non-histone. Okay, and this is how the chromosomes are formed or appear in our nucleus. Let me expand this. Yeah. Look at that. Short region of DNA double helix will be appearing like this. We are picking up only one portion of DNA. And beads on string forms chromatin. We can see the round structures, just like rudraksha, beads. And remember, Rakesh, 2 nm, 11 nm, 30 nm, these are nanometers. So you can appreciate how much depth they have gone, the research scientists, okay? And this 30 nanometer chromatin fiber of packed nucleosome will appear like this. And then section of chromosome in an extended form. This is how the only small portion, how much? 31 nanometers look like. See, you must appreciate this. Though we are taking the 300 nanometers length chromatin fiber, in that 30 nanometers only we are seeing. And then further, condensed section of the chromosome. All these beads are condensed, they become small, and in the electron microscope it appears like this. And finally, all these condensed form will be in the form of a chromosome during mitosis with the centromere, Rudraksha, in the center. So it appears like, when we see under normal microscope in our lab, you will see chromosomes like threads. But inside the thread, what will be there? We need electron microscope and advanced microscopes so that we can see all. So we are going into.
We can see this.
We can see this only under electron microscope, not under our compound microscope which we are having in the lab. Okay, Sandeep? And that is the reason why I always encourage you to go for higher studies. As you go into higher studies, taking molecular biology as your main subject, you will enjoy all this at university level, right? So chromatin, there are two types of chromatins. One is euchromatin. Euchromatin is loosely packed. Whatever we discussed just now, those structures are loosely packed, and transcriptionally active region of chromatin, it stains light. That means easily we can color it. Staining means when we apply some color, like I'm sure in 11th class when you came to lab, you were applying that methylene blue, iodine, methylene red, isn't it? For the tissues. So if the tissue takes that stain, the entire tissue will become colorful. Under the microscope, your slide will be very attractive. And you can see the internal parts that are present. Luckily, this euchromatin takes the color. Whereas heterochromatin, hetero as the name suggests, different. So it is densely packed, inactive region of chromatin. It is not active and it doesn't take color. It stays dark. So we can study DNA under this. And in my next class, I will tell you at least 1 or 2 experiments to prove that DNA is the genetic material. Okay? And I will post this in our WhatsApp group. You can go through at your leisure. Right? Take care. Bye.
Yeah.
Okay, sir.
Okay, take care. Okay, bye.
Good afternoon, sir.
Thank
Good
you,
afternoon.
sir.
Good afternoon.